A pleasant day, everyone. For today's episode, we're going to tackle DNA profiling, its significant impact to criminal investigation. Impartial fulfillment for the subject SciEd Bio 705. I am your host for today, Miss Eliza Micah Cruz, submitted to Dr. Chris Pastor. Let us now begin. Couple of decades ago, before different biological technique was used for investigation purposes, detective and police used skillful investigation, interview technique, evidences, and people testimonies to solve crimes. However, what if an innocent person is suspected for a crime that he did not commit? It will be difficult to remove the stigma of being convicted. That's why authorities as well as law enforcement must conduct thorough investigation and think of other way beside witnesses, testimonies to prove a person innocence. Can you think of other way to figure if a person is guilty for a particular crime? Some of you might think of using different biological advancements such as DNA profiling. DNA profiling from dramas, just like Partner in Justice, showed us that the process requires collection of samples from the crime scene. Watching Soko gave us the idea that DNA profiling helps in conducting investigation with ACE and pin down the real culprit in a more objective method, avoiding miscarriage of justice. While CSI give us a hint of how DNA profiling is being performed. Indeed, crime in different medias gave us an abundant knowledge of what DNA profiling is. But how is DNA profiling done? And what is its substantial implication in the society, legal system, health system, economy, and environment? Let us answer the following question as we progresses. But to further understand today's topic, we have here the following terminologies. Deoxyribonucleic acid, or DNA, is a molecule that contains the biological instructions that make each species unique. DNA is found in every cell from our body, from hair, teeth, bones, blood, and skin. All humans share 99.9% of their genes. The remaining 0.1% differ us from everyone else. And this unique fragment of DNA is organized within the 23 pairs of chromosomes in the nucleus in each of our cells. Chromosomes contain junk DNA, and this type of DNA is not involved in protein production and have no significant impact to one's health, but could be used for matching purposes. STR, also known as microsatellite, is the small segment of DNA or a string of repeating nucleotides unit which make us unique from other individuals. It is also known as one of the initial elements for DNA profiling. DNA profiling is the analysis of DNA variation for genetic identification and a method used in constructing genetic profiles which can be achieved by analyzing individual genetic marker such as the aforementioned STR. Development of DNA profiling can be traced back in the year 1984 when a geneticist named Alec Jeffrey observed a coded peculiar pattern occurring in his X ray film image of DNA samples. The first breakthrough of DNA profiling was in 1986. Initially, a suspect named Richard Buckland was arrested and confessed to the second murder. However, after genetic profiling was conducted, it revealed that the DNA extracted from the two murdered girls did not match the suspect, and because of this, he became the first person to be cleared of suspicion on the basis of DNA profile. The case almost went cold. However, in the year 1988, A local baker named Colin Pitchfork boasts about using a DNA sample of his friend in his place. When his DNA was reassessed, it was a match. DNA profiling involves a sophisticated steps which can be narrowed down into six. The first step is the extraction of cellular samples from the crime scene. The second step involved breaking down the cell to release the genetic material. The step could be accomplished by bathing the cells in hot water with salt before placing it in a centrifuge. After DNA extraction, fragmentation will take place. The step includes addition of restriction enzymes which recognize specific DNA sequence, breaking the DNA into shorter fragments. The fragments will be then placed in the well within the gel matrix, floating in a buffer solution before subjecting it into electric current. DNA fragment will then move across the positive side of the gel matrix. Larger fragments travel slower as compared to smaller fragments. As an output, DNA fragments organized according to length will be obtained. The next step include performing the Southern blotting technique. Proceeding to the final step, we have matching. Through the process, we can now view DNA band pattern and match it with another DNA pattern. Usually, FBI use CODIS system which analyzes repeated sequence of nucleotide in 13 loci of the chromosome. Using the CODIS system, it enables investigators to determine offenders that can be reflected in the Beercam DNA case and Golden State Killer case. Let us now proceed with the advantages and implications. Through the use of DNA profiling, crime rate had been reduced. DNA profiling could also be used to aid investigation and reassess old cases that went cold. The aforementioned method is also helpful in establishing familial relationship which is accepted by the court system for custody, child support, as well as inheritance. Other than the stated advances, DNA profile can also be used to identify genetic disorder and provide early and proper remediation. DNA profiling also help in fighting against food fraud. Genetic material of food samples can now be analyzed to identify if a particular food is substituted, adulterated, mislabeled, or underwent genetic modification. Through DNA profiling, biological community can be conserved. The method could be used to monitor geographical spread, genetic diversity, as well as promote legal justice associated with animal abuse. On the other hand, limitations and ethical issues that may arise on the method primarily focus on DNA privacy and an individual autonomy to share their DNA account. The first limitation is that DNA data might get into the hands of insurance, loan companies, or other institutions who could determine a person's predisposition to a particular disease. In line with this, storage of DNA profiles can be seen as an invasion of privacy and could possibly be a real threat, especially to those criminals who wanted to fraud their DNA. Scientists at Nuclex, an Israel company, can access multiple DNA profiles from databases and could manufacture DNA samples without obtaining any cellular material directly from the actual person, which is alarming and can give offenders idea on how to cover up their crime. Another limitation is that DNA from crime scene is prone to contamination, yielding false evidence or accuse an innocent person. The former limitation can be reflected in the touch DNA related scenario. A concrete example of this is the Lucas Anderson case, in which Anderson was arrested for murder after his DNA was found under the fingernail of a homicide victim. According to the investigation report, he was the same paramedic who responded to the murder. From this, it can be assumed that Anderson's DNA transferred to the victim while responding, and due to this reason, the charge was dismissed. Lucas Anderson case showed us how DNA contamination can impact an investigation, also set a good reminder to responders or paramedics to handle victims and evidences with great care. That's all for today, everyone. Hoping you learned a lot from today's topic.